Hey guys, I hope everyone is doing well. In this video, we're going to be talking about chromosome mutations. There is an outline and study guide for you to either print or open on Google Docs for you to fill in as we go through this information. So a mutation, let's start with just what is a mutation. So mutation is any sort of permanent change in our genetic material. So there's multiple things that might cause a mutation, but it's a change that is not reversible. Our cells do a pretty good job of checking and fixing mistakes, but this is a change that is not fixed. So this could occur in somatic cells. These are our body cells and therefore not passed to your offspring, so maybe in your skin or an organ. Or it could occur in your gametes. If it occurs in the gametes, then it's passed to the offspring, and then the offspring results with that mutation and that change in the genetic information. So chromosome mutations are specific to the chromosome itself. It's a change in the structure of that chromosome or the number of that chromosome. So if we're supposed to have 46 as humans, and we accidentally have a mutation that causes 45 chromosomes or 47 chromosomes, that would be a change in the number of chromosomes. Or we might change the format of the chromosome itself. So we might have a loss or gain of a chromosome, we might have a loss or gain of a piece of a chromosome, or a change in some piece of the chromosome, as we will see. So we're going to see 5 types of chromosome mutations. I would caution you to take a look at their names because the names are pretty descriptive and they pretty much help describe what is happening with that chromosome. So notice the term deletion, inversion, to think of to invert, duplication, think of to duplicate. Translocation, that one's a little more tricky, but trans means to move and location would be it's a position. And then insertion, to insert a piece. So we'll go through each of these. Let's start with deletion. So in the case of deletion, a piece of a chromosome is broken off. That'll be this yellow piece in this chromosome on the left. And then that piece is most likely going to be lost as the cell is moving around and dividing into new cells. So notice the new form of that chromosome is missing that piece. That is simply a deletion. Inversion is if that piece that breaks off then flips and then reattaches. So a piece of a chromosome, once it's broken off, flips around and reattaches. So it's pretty much like trying to read backwards, which does not make sense to the cell. Duplication, just as it sounds, is when you have a piece of a chromosome that is copied twice and therefore duplicated. So it's too much information for the cell to process. So a piece of the chromosome is going to be read, and when that chromosome is read, it's read twice accidentally, and therefore we have a duplicated sequence. This might be a gene or multiple genes that are duplicated. Translocation. So now we have two chromosomes interacting. It could involve multiple genes. So a piece of a chromosome breaks off and then reattaches but to a different chromosome. So it's going to be a little bit like crossing over, but in this case it's not homologous chromosomes. And therefore, it's information that the cell has a trouble processing because that gene information is not in its proper location. So a piece breaks off but reattaches to a different chromosome. So this might happen from one chromosome to another, or it could be a trading of pieces, as you can see in this image. So again, this will be in non-homologous chromosomes. Technically, it could be homologous chromosomes that's not an even trading. All right, and our 5th chromosome mutation would be insertion. So insertion is when we have a piece break off of one chromosome and attaches to a different chromosome, but it inserts in between. So it's similar to translocation because we have 2 chromosomes that are interacting, but in this case, that piece that broke off of one is inserted in between a whole bunch of genes on the other non-homologous chromosome. So we're going to add that to a different chromosome. So those are 5 basic chromosome mutations that occur that would impact the structure of a particular chromosome. Here's a little image. This is our original chromosome. I don't know if it's going to play. All right, so that's supposed to be an animation that's playing for you, but it's not playing. So we'll just move on to how do we get extra or missing chromosomes. So you might remember when we did our karyotypes, if you completed your karyotype, you would have had an extra chromosome or a missing chromosome. Or in our karyotype discussion, you saw we could have an extra or missing chromosome. So how does that happen? That would be considered a chromosome mutation and is caused by what's known as non-disjunction. So chromosomes, as you know, are supposed to separate. And when chromosomes separate, we end up with, our chromosomes ended up in different cells. Non-disjunction is the failure of those chromosomes to separate properly. So this could be, think about anaphase of meiosis, when the two X's separate from one another to be put into different cells, and they fail to do that. So both chromosomes are going to go to one side, and neither copy of that chromosome will go to the other side. This is going to cause our gametes, or those reproductive cells, to actually have too many chromosomes if both went to one direction, or too few chromosomes if neither went to the other direction. And remember that having 46 chromosomes is very important for humans, or whatever chromosome number that species has matters very much so. It's very important. Otherwise, it leads to disorders. So let's take a look at an image of this. So non-disjunction occurring. So our chromosomes are lining up in pairs here. This would be considered metaphase 1 of meiosis. And those chromosomes are supposed to separate, as you see here on the left. So notice the chromosome pair. One chromosome goes to one side, and the matching chromosome goes to the other side. In the case of non-disjunction, which you see here on the right, the two homologous chromosomes are moving to the same side of the cell, where neither of them is moving to the opposite side. So that's going to cause, in the second round of division, so we've jumped here to round 2, this would be anaphase 2 versus anaphase 1. Our new cells by the end, after telophase 2, end up with the wrong number of chromosomes. So since we had 4 chromosomes to begin with, we should end with 2 in each of these cells, because remember, chromosome number is cut in half. But in the case of non-disjunction, notice that 2 of our cells have 3, and 2 of our cells have only 1 chromosome when they should, in fact, have 2 each. So that's a result of non-disjunction, and it will cause the new zygote that is formed, if any of these cells is used in fertilization, to have too many or too few chromosomes, and therefore a disorder. So what might that look like? So in a karyotype, now I'm not sure how many of you were able to finish your karyotypes before we left, but karyotypes, if you remember, should have 2 of each chromosome if we're talking about a cell that has is a body cell, so therefore it has 2 of each, and that's called a diploid cell. But notice down here at the bottom, we have 3 of chromosome number 21 instead of 2. So that causes a very specific disorder. Having an extra chromosome or a missing chromosome, it very much matters where that chromosome is extra or missing, because it leads to very specific different disorders. So if we were talking about chromosome 21, that leads to what's called Down syndrome, which is a disorder that we are relatively familiar with because it's somewhat prevalent in our population. But if we were talking about chromosome number 13, that would cause a very different impact. Notice that having 3 of a chromosome is called trisomy up here at the top. So this is specifically trisomy 21, or 3 of chromosome 21. Here's another example. If this was to happen at our sex chromosomes, so individuals who are XX are female and XY are male, but look down here at the bottom, we have 3 sex chromosomes, XXY. This individual has what's called Klinefelter Syndrome. And the opposite of Klinefelter's is pretty much Turner Syndrome, not in terms of their symptoms, but in terms of how non-disjunction is occurring. In this case, we have only 1 sex chromosome, and that would be an X chromosome. There's no other sex chromosome, there's no second X, and there's no Y. This leads to what's called Turner syndrome. This is also called monosomy because there's only 1 of that chromosome. So you could have monosomy in a different location. It just happens to be at the sex chromosome for this particular example.